So this tutorial is going to review how to create information on an individual study. So the previous tutorial discussed the overall investigation. So within an investigation there could be one or more studies. And the study provides information on samples, their characteristics, and also any treatments applied to those samples. So having created the high-level investigation information, we're now going to create a study information. So there could be an identifier for the study. You could also provide a title to the study. And again, you could also provide any abstract information, as well as fill in information such as the funding source, funding agency, so a study can have one or more assays, and an assay is basically you use one of the samples that you define with certain technologies such as transcriptomics to generate data. So in this case, we're just going to create a study. So what I just did there was to click on this Add New Assay button. And what we're seeing here is a series of templates that we could then fill in with the information. So I'm going to look for the transcriptomics template, transcriptomics profiling, DNA microarray, from Affymetrix. I'm going to add that assay. So in this case, there's one investigation, one study, and one assay. But there could be any number of assays performed on the sample. Again, if there's any publication information that can be provided, this version of ISA Creator comes with some predefined factors that can be used, such as compounds, dose frequency, sample time points. Given that we just selected an assay, the transcriptomics profiling, what you can also do is you can see that a series of protocols for sample collection, RNA extraction, labeling. And these protocols need to be linked to actual protocols on the TOXBANK data warehouse. And this can be done by clicking on this study protocol type. And what we're going to do now is to search the TOXBANK data warehouse for either a number for the protocol number, or you could do a text search. And what it will do is it will bring back all protocols in the TOXBANK data warehouse that match it, and allow you to link each of those steps in the transcriptomics to an actual protocol. So here it actually found 20, and what you can then do is, if we open up here, we can look through the protocol list and select the protocol which matches the specific step for each of the protocol steps. So you would do that for each of the protocol steps. And again, you could also add study contact information. The next step, though, is to create information on the study. Those are the samples. So what you would need to do is to say, what was the source material? So first we need to know the source name, for example, hepatocyte. Medium. And then you can fill in various characteristics of the medium, the cells, for example, the organism. What we would do by clicking on that, double-clicking on that button, what we're going to do is search ontologies. So I'm going to look for, these are human samples. We do a search of ontologies to find terms, for example, in the NCI, you can see this has been defined, Homo sapiens, and we can link that term If there's characteristics about the sex or the strain or the age, those can be added. We can add things such as the organs. So if I do a search for liver, we can link it to the liver, tissue, if it's a specific cell or cell line. In this case, it was a hepatocyte, human hepatocyte. Again, we can link it to an ontology term. In this case, this was from the Ontology of Biomedical Investigations. You could also add if there's information on technical replicates. So in this case, there were 3 technical replicates. There could be 2 or 3 or however many. So we could add 1, 2, 3. The compound was doxorubicin. So here again, I'm going to look at this up in the ontology. And it's important when compounds are being used as treatment to actually use the OpenTox structure ontology if it's available. So here it's OpenTox, we have for doxorubicin. Let's select that compound. So as we go towards the right, you can find things such as dose. So we want to be able to then type in different doses. So in this case, it was done at 4 different doses. There was control, dose 0. So this we have control at dose 0. And everything was in micromolar. So I'm just going to click on the units. And the unit ontology is particularly useful for any unit to use to link to any unit. So there were 3 technical replicates, 1, 2, 3. Just going to copy this. So doxorubicin in the 3 technical replicates. Again, it was all the negative control, so all at 0 micromolar. You can then fill in other things such as the time point. So they were all done at 8 hours and 24 hours. So for example, these 3 were done all at 8 hours. And again, we need to link hours to the ontology. Here, the unit ontology, hour. Copy those over. So what we want to then start to build up is the full matrix of different doses, time points, and each row of this table will then refer to one treatment. So for example, here we did it 3 technical replicates at 0 dose. We could also repeat that for a dose 0.4. And again, these are all done at micromolar. And then you can see how we can start to build up a table showing all the different combinations of dose, time it was treated, so that each one is a different sample. And then the final step is to then type in a name which reflects that particular sample. So in this case, we'll type in the name TG Gate in vitro. This was testing doxorubicin. It was the control at 8 hours, technical replicate 1. And again, you can repeat that for each of the different sample treatments that were done. And then this is the starting point for where we define the assay, because each assay will then perform an experiment on each of these samples. So to summarize, we've just created a single study and we've defined the characteristics of the cells, the treatment, as well as the time points it was treated at and the dose levels. And the next tutorial will now cover how we implement an assay.